Yeah, good afternoon, everyone. Is there someone on the other side? Let me know if there is a voice coming from the background. Some music is being played in our society. So is the voice coming on that side?
No, ma'am, I can't hear any background noise.
It's okay, na? ठीक है, चलो,
Yes,
fine.
yes, yes. Okay.
Thank you. Yeah, good afternoon, students. So, till yesterday, we are done with the chapter on reproduction, including the human reproduction and the plant reproduction. Also, we talked about in detail about the reproductive health also, okay? Now, the next chapter that is related to the genetics part, and again, a very interesting chapter this is, and where we talk about different kinds of the laws which the Mendel gave, okay? How did the Mendel work on the pea plant, okay? What different type of laws given by the Mendel? What are different characteristic features he has studied in the plant? And accordingly, what are the different laws of the genetics, okay? So just a preliminary, you can say the knowledge given to you regarding the genetics. But obviously, we'll talk about both the inheritance of the one gene as well as the inheritance of the two genes together. Means the monohybrid and dihybrid cross we are talking about, okay? And then we'll see how to complete a Punnett square. How can that be done? How the laws, different laws, Mendel's different laws can be understood, okay? And then obviously, as the time passed, the Mendel's law was restudied and how it was reinvented with the new laws into the picture, okay? So there are many things that Mendel's or whatever law given by the Mendel has not been completely followed by all the genes. So we talk about the variations also happening over there. Okay. Fine. So let us continue. Let us continue with the chapter today. So if I talk about the genetics part, let us just first introduce you to what the genes are. Okay. So as soon as I use the word genes, as soon as I would use the word genes, obviously you think about something that is called the DNA that is located inside the chromosome. And within the chromosomes, you have these DNA segments, and these DNA segments are like the library of the information. So, what will be the skin color? What will be the eye color? What will be the hair color? What will be the height of the individual? Everything, every small little bit of details are there in this DNA. And the part of the DNA that is holding such kind of information, in general terms, we use the word genes for them. Okay, fine. So if I talk about the way genetic study has been done, okay, we definitely first talk about Mendel's law. Now, it is very important for you to understand that how did Mendel came to certain conclusions, okay? So before that, let us discuss about what Mendel's work was, okay? So Mendel talked about he studied actually on a plant that is called Pisum sativum, that is known as, right? That's a common name. That's a biological name given to a pea plant. The Pisum sativum I'm talking about. That's a sweet pea I'm talking about. Okay. And in this sweet pea, Mendel was actually working in his backyard. Okay. He was growing this many number of plants in his backyard. And obviously, these were the plants which used to show the desired characters or the desirable changes within a year. These are all annual plants. These are okay. So it was very helpful for the Mendel to actually understand to get the results very quick, and also these results were very dramatically different from each other. So what are the characters? What are the phenotypic characters we talk about? What is the phenotypic characters did Mendel study? So there were 7 different type of characters which were studied by Mendel. At times, CBSE does ask you only the questions that are related to what are the characters studied. Okay. So, and among them, which one is a dominant and which one is a recessive? That's also been asked in the examination. So, this particular table in front of you is going to be helpful. So, what are the characters? Means, what are we talking about? We are talking about the phenotypic character we are talking about, like phenotype. Phenotype is the external character that is visible to us. We talk about the phenotypic character. So, like for example, the seed shape, whether the seed shape is round or a wrinkled one. Then he talked about the seed color, whether it's a yellow or a green. Then talked about the flower color, whether it's a violet or white color. Then a pod shape, whether it was a constricted pod or a full pod. And the pod color was there. You know what the pod is, inside which these you can find these the seeds of the pea. That's called the pod. Okay, the green or a yellow. And then obviously he talked about the flower position also. If it's an actual flower position, then it's obviously it's on the somewhere on the axis of the main stem, and if the terminal means that it's somewhere on the end of the that particular stem. Okay, so obviously depending upon different type of character, now we know that which one is the dominant, which one is recessive. At the time when the Mendel worked, look at the year in which Mendel worked. Okay, it was around the year 1856 to 1833. Obviously, during this time, there were no microscopes where there hardly any information related to the genes or the DNA was there. There was no information how the cell is working. So Mendel's work was just the theoretical, you can say the observations he has put up, okay, of the multiple experiments which we have done, which he has done. So later on, once this all work was done, he has his work was once again you can say it was re-established once again. It was redefined once again. There were many scientists who actually once again have gone through the results what the Mendel has given and again redefined everything. Okay, and that was in the year around 1900. So obviously, if I'm talking about the Mendel's work, first I'll talk about the Pisum sativum. That's the pea plant on which he worked. Then I'll talk about the phenotypic character. That's seven in number. What were the seven different characters he studied? Okay, and among them the few were the dominant one and the other few were the recessive one. Okay. So remember that such questions are also the stem height, also like a tall and the dwarf one is there. So out of these experiments which he was doing, okay, there are certain terminologies we would like to share with you before going ahead with the experiment so that you are able to understand that exactly what Mendel did. I'm opening my whiteboard in front of you. So if I'm talking about the Mendel's work, okay, it is something which is known as a true breeding, right? Something which we call as a true breeding. What is the meaning of this true breeding? Suppose, for example, now we know what it is. Okay, so it should not have some kind of a hidden gene. For example, if suppose it's a tall plant, it is, right? And if it's a dwarf plant, it's a tall plant and it's a dwarf plant. What is the meaning of a true breeding plant? It means that the tall will have both the genes on both the alleles as capital T, and dwarf will have both the genes of a dwarf gene on both the alleles. Okay, you know what the alleles are? The pair of genes that are present on the chromosomes. Okay, so these are known as true breeding genes, and true breeding genes were self-pollinated at times. They were crossed with each other. Like for example, in front of you, I've given what is this tall? This is a phenotypic character it is. What is it? Capital T. Capital T, it's a true breeding, and so it's a genotypic character it is, okay? So always whenever you are putting a cross, make sure that the first step you write is a parental phenotype you're writing, okay? Then you write a parental genotype as I have written. And the next step will be from the parental genotype, you see how the gametes are being formed. So always put gametes inside the circle like that, inside the circle like that. For the dwarf variety also you have the gametes formed. Put them inside the circle like that. Okay. And once these are crossed with each other, you get the next generation as capital T, small t, something like this is there. And these all plants are tall. So in the F1 generation, what is that F1 generation? The first time the parental population is crossed with each other, you receive a F1 generation. And this F1 generation, F stands for the filial, F-I-L-I-A-L. That's a filial generation we talk about, right? So what is this filial generation? It is the first time when the parents are crossed. Okay, the first generation obtained from it is known as a F1 generation or a filial generation, right? So when Mendel started working on all these things and he was using a true breeding plant, so when he crossed tall plant with a dwarf plant and he in the F1 generation, he got all tall plants, okay? So he came up with a law that is called the law of dominance, that is known as a very important law, except few of the, you can say, anomalies. Every gene follows this law, okay? That is called the law of dominance, this is known as. And the law states that out of the two characters, right? The character which is represented in the F1 generation is the dominant character, right? And the character that is hidden, if you can see, this is a tall plant, but it has a gene small t also. What does that mean? That character is hidden because you can see all the plants as tall. So in F1 generation, you have one, you can say, one character, one phenotypic character that is visible. So the character that is visible in the F1 generation is the dominant character, and the character that is hidden in the F1 generation becomes a recessive character. Understood? So that is a law of dominance, and it holds good on like 90% of genes. When we talk about later on, when it was founded, there are certain genes which do not follow the law of dominance. We'll come to it later on, but most of the genes are following the law of dominance. Okay, so we have once again, I'm repeating, this is a cross being made between tall and dwarf plant. The tall is a homozygous tall. Again, I'm using word homozygous. Let me explain you what is the meaning of the term homozygous. There are two terms being used. One is a term called homozygous, right? And another one is a term called heterozygous. Heterozygous, right? So how do you define a homozygous and heterozygous condition? So if in a chromosome, both the alleles are carrying same type of gene, but it is a capital T, or suppose it is a small t, it is okay. Such type of combination is known as a homozygous characters. It is no homozygous genes, these are known as. And if suppose both the chromosomes are carrying different type of genes like capital T and small t, okay, that becomes a heterozygous condition that becomes, okay. So going back to what I was explaining to you. So true breeding means what? It's a homozygous condition for the tall. It's a homozygous condition for the dwarf. And when both the homozygous conditions are crossed with each other, you get what is called the all tall. But this is a heterozygous tall. This is okay. Initially, what you have taken in a parent was a homozygous tall, but now it's a heterozygous tall because there is one gene of a dwarf plant that is hidden. And in the F1 generation, you get only what is there in the what is visible to you. That's all tall is visible. Okay. Fine. How do we get to know that there is some gene that is hidden also? So whatever result you get in F1 generation, like capital T, small t, right? This is a generation you're getting in F1 generation. So that capital T, small t in the F1 generation will be crossed once again. Okay, this process is known as a selfing process. This is called the selfing. This is known as whatever result you got in the F1 generation, that is capital T, small t, you're going to cross with the same kind of a population. So the results of the F1 generations are crossed with each other, capital T, small t, and that is how from the F1 generation, you receive what is called the F2 generation. Okay. So once again, you will be crossing them with each other. If you want, again, you can be making the gametes also. So you have capital T, small t gamete over here, capital T, small t gamete over here. Again, you have a capital T and a small t gamete. Whenever you're writing the gamete, make sure you're writing inside a circle you're writing. Okay. There's one way of representing these structures, and not in this cross, but maybe in some higher categories of cross. It's very helpful. Okay. So like this, you can do. You have a capital T, small t on this side. You have capital T, small t on this side. Just make a cross. Okay. Capital T, capital T. You have capital T and small t. In this particular case, you have. And the last case will be capital T and small t. Understood? So if I look at the phenotypic ratio, if I look at the phenotypic ratio, right? And if I look at the genotypic ratio, if I look at the phenotypic ratio and I look at the genotypic ratio, the phenotypic ratio is 1 tall, right? There is one more tall, there's one more tall. There are 3 talls are there and 1 dwarf is there. So you have 3:1 is the ratio. Sometimes this ratio is asked in examination, okay? On the other hand, let us talk about genotypic ratio. The first one is tall, but it's a homozygous tall. The other two are tall, but a heterozygous tall it is. So you will put 2 over there separately. And the last one is a recessive one. So you're going to put it like this. It's 1, right? The phenotypic ratio is 3:1 and genotypic ratio is 1:2:1. When after doing the selfing with the F1 generation, we have crossed and we have received what is known as the F2 generation we have received. And in this F2 generation, we got a phenotypic ratio as 3:1 and a genotypic ratio as 1:2:1. Okay, fine. Now, since in this particular cross which I made, I took only one character at a time. Mean, I was just talking about either a tall or a dwarf plant. Okay, so actually I was talking about just one character at a time. So such type of crosses are known as a monohybrid cross. Remember this point also, right? So if I'm taking one character at a time, like for example, in this particular cross, I took one character, that is the height of a plant I took, right? I didn't discuss about any other character. I was just talking about a tall and a dwarf condition, that is just referring to the height of a plant. So if I, at one time, if I'm talking about just one type of character, I'm discussing only about one type of character, I'm mentioning only about one type of character, I said I'm talking about the monohybrid cross. So now you understood, in a monohybrid cross of the F2 generation, you have a phenotypic ratio of 3:1 and a genotypic ratio of 1:2:1. Okay. And every time you find this thing, especially in the F1 generation of the monohybrid cross, from that F1 generation, the Mendel concluded what is known as a law of dominance, which says that the character that is expressed in F1 generation is a dominant character, and the character that is hidden or suppressed in the F1 generation is a recessive character. And obviously, that recessive character was visible in the F2 generation, right? Because the gene of that particular thing was present, and obviously, it requires a homozygous condition to be expressed. Okay, small t, when in a homozygous condition, that condition will be expressed. Okay, so the gene was there, but it was hidden in the F1 generation, but it was you can say expressed in the F2 generation in the ratio of just 3:1. Okay, so that's a monohybrid cross and the law of dominance. We talk about both the things. I think is now understood by you. Let me get back to my earlier share screen. Fine. Now, if you look at this diagrammatic representation over here of a monohybrid cross, again, they have mentioned these things over here. And then we talk about whatever is I have made you understood, it's all given in this whole paragraph. Okay. They talked about the alleles, they talked about the genes, they talked about. Also, one more thing is that Mendel never used the word genes because he was not aware of the nucleus, he was not aware of the chromosomes. He was, during his time, there was nothing like a concept of DNA also. So he never used the word genes. Actually, word term used by Mendel was the factors. So somewhere when you're mentioning in your answers, or sometimes it can come as an MCQ also. So remember that he never used the word genes. He used the word factors. He said these are the factors that are influencing the character of a particular this thing. Okay. So then what are the characters to be taken? What is the genotypic, phenotypic character? What are okay? They have explained all these things to you. And finally, we come to the law. The law states that it is 1:3:1. The law, after preparing all that Punnett square, okay, we see that the law, the phenotypic ratio, as I told you, for the monohybrid cross is 3:1, and for genotypic ratio is 1:2:1. It is okay. So obviously, this was one way in which the first law was discussed. Okay. There was a second law also, the Mendel game from this monohybrid cross only. That also now we can discuss. So the second law states that, right, my screen is shared. So the second law states that it is known as the law of segregation. Second law is known as the law of segregation. Right now, what is this law of segregation? What we talk about it, okay? When I was drawing the structure of capital T, crossing with small t, at one time I said that let us draw a gamete, right? And just put it in a circle like that, right? Now, why I'm asking you to put it in a circle like that? Because the gametes which are formed, they are not dependent on each other, right? I'm saying this thing because in the next generation, like for example, it was capital T, small t in the parental population. But in the next generation, you did get one capital T and small t. This is a new kind of a combination that is not there in the F1 generation. It's not there in the parental population. Okay. What does that mean? That all the gametes are independent of each other and they tend to segregate from each other during the process of meiosis. Right? So meiosis में जब ये gametes बनते हैं, they segregate from each other, means they are not influenced by each other. They are not somewhere or the other. They're not going together every time. It's not necessarily that capital T should be having only capital T, small t should be having only T. There could be a combination of capital T and small t also. Even if the parent is not having that kind of a combination, still the F1 generation can have that kind of a combination also. Also, if I looked at 1:2:1 combination, 1:2:1 combination. What does this combination say? Capital T, small t, and what was the two number given to? Capital T and small t. This capital T and small t is not a parental combination, it is okay. So where did it come from? It came from the place that the genes or these segments or the gametes, they are independent of each other. They're segregated during the meiosis process. They are segregated and they don't influence each other's location. And you can have a good number of new combinations, even if that combination is not there in the uh, parental alleles. Okay, that can be there in a new allele also, and that is known as a law of segregation. So both the law of dominance and the law of segregation can be explained through a monohybrid cross. So sometimes during the examination, they ask you either only the law or they're asking you, or sometimes they ask you to show the cross and then derive the law. Right? So you should be ready for both the things. So in both the cases, I'm talking about either if I'm talking about the law of segregation, if I'm talking about the law of dominance, in both the cases, you can take an example of a monohybrid cross, show it till the F2 generation, and then explain, taking an example from the cross which you have made, what is the F1, what is the law of dominance, and what is the law of this thing. What you say? The law of segregation. Okay. So this is again an important thing. Again, let us go to one more thing. These are all known as today the laws were proposed by the Mendel are consolidated his understanding of the inheritance of the monohybrid process. Today, these laws are known as the principles of the law of inheritance. Okay, and obviously the first law is the law of dominance. What does the law of dominance say? They have given it in 3 points. Okay, just remember these 3 points. The law of dominance say that the characters are controlled by the distinct units known as the factors. He never used the word genes, remember. Factors occurs in pairs. Why was he saying factors occurs in pairs? Look at the chromosome. This is chromosome having capital T over here. And this is again something having small t over here. Always in a pair. And today we know that pairs are known as the alleles. These are known as, okay. He also said this thing. That the factors are always appearing in a pair. And also he said that the one member of the pair always dominates the other member, right? So out of the two members which are present in a pair, it will be the one member that is going to dominate the other member, and that is known as a law of dominance. Again, this can be explained through the monohybrid cross. Then second law, which again as stated, is the law of segregation we talk about. What is that law of segregation? That the homozygous, obviously you have the homozygous parents and you have the heterozygous F1 generation. What does that mean? The gametes are independent of each other and they tend to segregate at the time of this thing at the time of the meiosis process. Okay, that is how from homozygous parent you get a heterozygous individual. Okay, so remember both the laws, they come as a law of inheritance also. So the law of inheritance is this thing. Fine. Okay. There are a few other things also in between what the NCERT has discussed apart from discussing the laws of the Mendel. Okay. Okay, I'll open my white screen once again. Fine. So apart from using your principles of inheritance, okay, that is the law of dominance and the segregation, uh, the topic also talks about what is known as a test cross, right? Now, what this test cross is, for you to understand test cross, okay, see what's happening in a test cross. First, you should understand this thing, what's happening in a test cross, right? Suppose I have a parent that is capital T, and a parent that is capital T and small t, right? Phenotypically, remember carefully, both the individuals are tall. This is also tall. This is also tall. But there's a huge amount of difference between both of them, right? The first one is homozygous tall. The second one is a heterozygous tall, okay? So how do I get to know that the parent which I am taken doesn't have a hidden gene inside it, okay? Because going for any kind of a true breeding experiment, I should be knowing that all the individuals are the true breeding, means they all are the homozygous in nature. So both are tall for me. They appear both as tall, but I need to do some kind of a test to finalize this thing. It's a tall, and at the same time, it's a homozygous tall. Okay, so there's one cross that is called the test cross. Which identifies whether the parent is a homozygous dominant or it's a heterozygous dominant, because dominant conditions can be expressed in both ways. Even if it's a homozygous, it's expressed. Even if it's a heterozygous, it's expressed. So obviously, we need to confirm whether it's a homozygous or a heterozygous it is, right? So the test cross, if I'm talking about the test cross, I'll just mark a barrier in between. I'll explain you through two experiments, right? Actually, two crosses I'll explain you through. On one cross, I'll cross this homozygous tall with the recessive parent, that is small t, small t. On the other hand, I'll cross this with again small t, small t. So what are you doing in a test cross? The individuals are crossed with a recessive parent. Remember this thing. Why this test cross required for? This test cross is required so that I know whether the dominant conditions are a heterozygous dominant or they are the homozygous dominant. Okay. So let us make a Punnett square for this. The first one, let us see what the answers will be. And for the second one also. Fine. So you have capital T, you have small t, you have small t for the first one. Let us make the Punnett square for the second one also. Right? So you have like this, you have like this. Okay. Again, you have capital T. At this place, you have a small t, and you have small t and small t. So what are you doing? You are crossing both the individuals with a recessive parent. Okay. Let us see the result in the first generation. Capital T, small t. You have a capital T, small t. Again, you have a capital T, small t, and a capital. For the second one, you have a capital T, small t. You have a small t, small t. Then you have a capital T, small t, and you have a. In this particular case of the first test cross which we have done, all tall plants are obtained, right? While in the second case, it is actually 50% tall, right? And it is 50% dwarf, right? If I'm getting the result like the first one, where all my individuals are representing the dominant character, it means that I have taken my parent as a homozygous dominant, okay? If I am taking it, it is 50% tall and the 50% recessive characters are also visible after my cross being made, okay? It clearly shows that the individuals are not homozygous, but they are heterozygous tall these are, okay? So I think this is clear. This is known as a test cross. This is known as, and this type of a cross is meant to make you understand that whether the two breeding crosses which you are taking is a homozygous condition or a heterozygous condition. Again, the questions are being asked. What is a test cross? What is the significance of the test cross? And what is the result of the test cross? What is the result of the test cross? If it's a homozygous individual, you will get all individuals from a dominant condition only. If it's a heterozygous individual, you'll get 50% from a dominant and 50% from the recessive. That is how the test cross works. Okay, fine. I think this is also understood to you. Okay, let me get back to my sharing. Okay, so once this is understood, once the test cross is done and you have learned the two laws also, okay, let us come to the next thing that explains you about a law that is, you can say a condition, not a law, but a condition that is not following the Mendel's law of dominance, right? So the condition, I told you there's some kind of anomalies there. Mendel's law was like on hundreds, 100 years before, even now more than 100 years before. At that time, there was no. He has restricted himself only to one plant only. That's a pea plant. Okay, most of the plants around you do follow the Mendel's law. Later on, during the studies, it was found out that there are certain plants which do not follow this law. And what condition they do not follow? Let us see. Okay. So they've given a condition of a plant that is called snapdragon dog flower, it is known as, okay. Anthurium it is known as. So you have a dog flower, a snapdragon or anthurium, okay. And this flower is a good example to explain what complete dominance, okay. How do you make a cross? You start with the parental phenotype as told to you. The parental phenotype will have a true breeding variety. You have a red and a white color flower available to you. So the the parental population, P generation they have written, will have capital R or a small r, right? What will the gamete formation happen? It is the one is capital R, then you have a small r, and then you have a F1 generation. The expected F1 generation in this particular case was all red plants. If it is following a law of dominance, then in the F1 generation, you should be having all plants as a red color flower, okay? But what happens in this particular case is that you don't have a red color flower at all. Actually, you have a pink color flower in this case, a combination of both the white and the red. Okay. So F1 generation is covered with the pink flower. And then when you try to get F2 generation, okay, again, the phenotypic and the genotypic ratios are slightly different. In this case, instead of getting a phenotypic ratio of 3:1, you get something new ratio, and that is of 1:2:1. Right? So what is this 1:2:1? This 1:2:1 is a ratio of the phenotypic ratio of a plant where actually the new combination has emerged, and that we are given two number to it, and this new combination is related to the process of incomplete dominance. Okay, so why the dominance didn't happen? Because both the genes have expressed themselves in some or the other ways. It was a white gene also that was expressed, it was a red gene also expressed. It was expected that only the red gene will be expressed. That didn't happen. So it is a combination of both the genes that was there, and this dog flower was having a pink color flower that was absent in case of the parental generation, right? This is an best example to explain the law of incomplete dominance, which states that both the parents, the combination with the allele which are present in both the parents are not going to be visible in the F1 generation. Something new will come up, okay, in the F1 generation. So remember the example. And whenever such questions are asked, make sure you're making a cross also, and you're explaining it nicely through the all the diagrammatic representations and the crosses and all, okay. So this is the law of incomplete dominance. Fine. Then there is one more concept that doesn't you can say abide to the law of dominance, okay? And this is known as a codominance. Literally, this is known as, to understand the process of the codominance, we need to understand slightly the blood groups of the humans, okay? How the blood groups of the humans are expressed, the genes of the blood groups of the humans are expressed. We need to understand that thing. So if I'm talking, you all know that there are four different type of blood groups of the humans, okay? You have four different type of blood groups. You have A, you have B, you have AB, and you have O, right? So obviously, which is going to be the one which is A blood group? It is going to have either a combination of IA, IA, or it is going to have a combination of IA and IO, which is a recessive gene. In both the cases, you have a blood group O, right? Sorry, blood group A. In case of a B, you have either IB on both alleles, right? Or you have a combination of IB and IO, right? Again, in both the conditions, you are having B as a blood. What happens in case of an AB blood group is very interesting, okay? You have a combination of IA and IB, right? It means that in this particular case, neither of the genes are dominant, neither of the genes are recessive, right? What is this condition? This is the condition where both the gene A carrying and both the gene which is carrying B gene, they both are equally dominant in nature, right? That is why in blood group AB, you find both the type of expression of the genes A also and B also, right? So what is it is having in both the alleles, it is having A also and B also. So instead of one gene getting dominant and getting expressed as an antigen over the RBC, it is actually both the genes, right? A also and B also that is getting expressed. What does that mean? The two genes together over the alleles are dominant. Both the genes are dominant. So this is a condition, what you call it as a co-dominant condition, means both the genes are dominant. And that is how you get a blood group AB in which both the genes A and B are dominant with each other. So the example of the incomplete dominance, if sometimes it is just asked that give any two conditions in which the law of Mendel's law are not followed, right? It's not, they don't ask you the direct question. Sometimes they just ask you that give the conditions under which, explain the conditions under which the law of Mendel's law is not followed, okay? So you can always give these two conditions. One was incomplete dominance, where we talk about a dog that we talked about, okay, in which the combination of red and white was creating a pink color flower, right? So that red and white, that pink was neither in any of the parents, okay? But you did get in F1 generation. That's an incomplete dominance. Slightly, both the genes are slightly showing their character and that giving up rise to something new character, okay? On the second hand, if I talk about the codominance gene, the codominance genes means both the genes are expressing themselves equally, right? It is not a partial expression of any of the genes. Both the genes, under their 100% capacity, they're expressing themselves equally. That is how the RBCs of the AB blood group, right, will have both the gene antigen having the A also and antigen B also. How do they get antigen A and B? Because both the genes IA and IB have expressed themselves, and this expression has led to the blood group AB, where the RBC is having both the type of antigens on its surface, that is the A antigen and the B antigen. Okay, so this type of a condition is known as a co-dominant condition. This is right. So, um, So in today's lecture, what we studied, something very interesting facts we have studied. We talked about the Mendel's law, and we talked about the incomplete. We talked about test cross also. We talked about the codominance condition. We talked about the incomplete dominance. See, at times they were like what they have done for explaining the codominance condition. They've given you the complete list of blood groups they have given you, and what are the different type of genes that are what are the different type of genes that are responsible for creating such type of group. It is not necessary to write each and everything. Suppose it's a 2 mark or a 3 mark question. No need to give a complete table. Just give talk about the IA, IB, and how the both the genes are having both the things in them. A and B genes are there, and both the genes are expressed and equally expressed. They are 100% expressed both the genes, and that is how we say that it's a codominant condition, right? Okay, so right. In the next class, it's time is almost up. Okay, in the next class is again an interesting factor of the multiple alleles we talk about. Or let me just discuss this thing with you of the multiple alleles. See, if more than two genes are responsible for one character, right? At times I'm saying capital T and small t for a height of a plant. But in case of the blood groups of the humans, it is also responsible for the multiple allele. Like IA is a gene that is responsible for the blood group. IB is a gene responsible for the blood group, and IO is also the gene responsible for the blood. It is more than two alleles that are responsible for the human blood group. Such type of conditions are known as a multiple alleles. These are known, right? This is the condition of a multiple allele, where more than two type of genes are responsible. So see, if they're asking you the questions related to the multiple alleles, do give all the details of all the alleles with the blood group. And right in the towards the end, that it is more than two genes actually that are responsible for the formation of the blood groups or the antigens on the surface of the RBC. And that is why we say that it's a concept of a multiple alleles. So blood groups are following both the concept, the codominance also, and the multiple alleles. In both the cases, you can always explain with an example of the human blood group. Okay. I think we can wind up now today. Tomorrow we'll discuss about again good Punnett square. A larger Punnett square we'll be discussing about, where we talk about the two different type of genes we'll talk about, and how the inheritance are there in these type of genes, and what is the phenotypic and a genotypic ratio, and few other things related to the Mendel's law of genetics. So, really, thanks all of you for joining. See you all tomorrow in my lecture. Thank you.